Good morning, grade 8 students. In the previous video, we finished this activity. Today, I'm going to apply what we studied in an exercise. Please pay attention. A small revision about cellular metabolism. It is the sum of all chemical reactions that take place in the cell. It consists of the series of chemical reactions of synthesis, assimilation and degradation, oxidation. Look at the picture. When the cell consumes oxygen gas and glucose, it undergoes oxidation to produce energy and carbon dioxide. This energy is used by another mechanism called assimilation to produce organic matter. So the cell consumes fatty acids, glycerol in the presence of energy to produce lipids, or it consumes different forms of amino acids in the presence of energy to produce proteins. Let's start the exercise. During the assimilation mechanism, the cell synthesizes new matter such as proteins. To identify certain elements necessary for assimilation, we perform an experiment on skin cells of a rat. Do you remember what are the essential elements needed for assimilation and the synthesis of proteins? Amino acids and energy. Yes, this is true. We place these cells in a culture medium containing radioactive amino acids or labeled amino acids. We add to this medium glucose, which is a nutrient utilized by the cells to produce energy. Okay, here they mention the process of oxidation. They are saying that glucose is essential for the production of energy. Few days later, radioactive proteins are formed in these cells. We all know that radioactive proteins are formed of radioactive amino acids. Part A. Compare the culture medium and the cells at the beginning and at the end of the experiment. So we must compare the constituents of the cells and the surrounding medium. So at the beginning of the experiment, we can see that the culture medium contains radioactive amino acids and glucose, while the cells do not. What happens after a few days? We can see that radioactive amino acids and glucose disappear from the medium. However, radioactive proteins are formed inside the cells. So when you want to compare the culture medium to that of the cells at the beginning of the experiment and at the end of the experiment. So, at the beginning of the experiment, the culture medium contains radioactive amino acids and glucose while the cells do not. However, at the end of the experiment, the radioactive amino acids and the glucose disappear from the culture medium while the cells contain radioactive proteins. Now, the second part of this question, derive the origin of the formed radioactive protein. So the origin of this protein is radioactive amino acids. These proteins are formed from the radioactive amino acids that were in the medium. What about glucose? Cells use glucose to produce energy because without energy, assimilation cannot occur. The production of protein without energy cannot occur. In addition, we measure the amount of energy consumed by these skin cells and the quantity of the formed radioactive proteins. The results of the measurements are given in document 2. So let's see the table. We have the amount of energy consumed by the cells, 0, 1, 2, 3, 4. So the amount of energy is increasing. And what happens to the quantity of the formed radioactive protein? Let's see the numbers 0, 4, 8, 12, 16. So the quantity of formed radioactive protein increases with the increase of the amount of consumed energy. Draw a graph line curve showing the variation of the quantity of the formed radioactive proteins as a function of the amount of energy consumed by the cells. First of all, you must know the variables on x and y axis. Do you remember? The variation of y as a function of x. So the variable on the y-axis is the quantity of formed radioactive protein. However, the variable on x-axis is the amount of energy consumed by the cells. After that, choose the correct scale. X-axis 0, 1, 2, 3, 4. The difference is 1, so we can take that every 1 centimeter represents 1 AU, 1 arbitrary unit, because on the x-axis we have the amount of energy consumed by the cells. And directly plot it, so 0, 1, 2, 3, 4. What about the numbers on the y-axis that represent the quantity of the formed radioactive proteins in microgram per milliliter? 0, 4, difference is 4, 4, 8, difference is 4, 8, 12, difference is 4, 12, 16, the difference is 4, so we can take that every 1 centimeter represents 4 microgram per milliliters. And directly plot it, 0, 4, 8, 12, 16. Now, when the amount of energy is 0, the quantity of formed radioactive protein is 0. Put the point on the graph. When the amount of energy consumed by the cells is 1, the quantity of the formed radioactive protein is 4. Plot it on the graph and continue plotting the curve. Do not forget to write the scale and the graph. The title of the graph, the variation of the quantity of formed radioactive proteins as a function of the amount of energy consumed by the cells. Part C. Analyze the results shown in document 2. Let's analyze the table. When the amount of energy consumed by the cells is 0 AU, the quantity of the formed radioactive proteins is 0 microgram per milliliter. As the amount of energy consumed by the cells increases to 4 AU, the quantity of the formed radioactive proteins also increases to 16 micrograms per milliliters. Part D. Based on the information provided by documents 1 and 2, indicate the elements necessary for this assimilation. In document 1, we showed that radioactive amino acids are essential for the formation of radioactive proteins, because when radioactive amino acids disappeared from the medium, radioactive proteins are formed inside the cells. You can check back document 1. However, in this document, we showed that the energy is essential for the formation of radioactive proteins. Without energy, the cell cannot undergo assimilation. So the necessary elements for the formation of proteins are amino acids and energy. Thank you and see you soon.